alpha copper trafficking proteins, you come up with about 100, about twice as many iron proteins, and at least 10 times more zinc proteins. And remember, the zinc and iron are usually balanced in the cell. Zinc is present, and it's used for catalytic chemistry, where the protein binds 3 points and then leaves the site open for other molecules to come in and be organized and undergo chemical reactions at the metal surface. This has captivated most of our understanding of zinc biology over the years. This has been shown with discoveries by Aaron Kluge and co-workers and another team at Yale of zinc sites that were not doing anything catalytic. And they had 4 ligands, these known as zinc finger proteins. And Jeremy Berg really opened up the field when he told us that it isn't just a couple. There are hundreds, if not thousands, of zinc finger proteins encoded in genomes. Okay, and these structural zinc sites, so-called structural sites, are involved in transcriptional control. They're involved in protein degradation, the ring finger proteins, and the E3 ligases, and they're in almost all polymerases, and almost every type of ribosome and every type of organism has a zinc dependence. But the zinc is not. What is it doing there? And so this is one of the questions that have been bothering me for a very long time. Like described it as a zinc metalloregulatory model. Is nature using zinc occupancy to control the functions of these types of proteins? Is it a type of master switch? And it could maybe selectively activate groups of zinc proteins. So I had this question, and this is what's led us to measure zinc in fluxes in many cells. And I asked my collaborator, who's a follicle biologist. She studies the maturation of the human egg and the hormones that drive follicular genesis. And she happens to be my wife. And we were walking on the beach one day, and I said, why is it that the sperm head has so much zinc? Could it be that the sperm delivers the zinc as a signal with the genetic material to wake up an egg that might be asleep for 40 years in a human? And she looked at me in the way that only a spouse could do, and she said, I don't care. How can you not care? Zinc could be the missing link in a whole suite of different types of mysteries of how cells operate and make decisions. And I was about to make the case even more fervently, and she said, no, it's not zinc I don't care about. She said, I don't care about sperm. And when she showed me this picture, she said, this is the largest cell in the human body. It's over 100 microns in diameter. And one is born with every egg that shall ever ovulate, and they're present at birth, and they're selected and matured, and in fact, there's no cells that take care of them up until the point of ovulation. And these are the smallest and most insignificant cells in the body, these little things moving around the sperm. She said, does it look like they know what they're doing? She has a gamete problem, and we're trying to get that taken care of. But she told me that if I told her there was something going on with zinc in the egg, that we could talk, and that was the end of our conversation. So what happened is, I want to set this up to show you the young woman that came in the lab and took on this challenge. But before I do, I want to help you understand how important these temporal fluxes of metals are. And some of you are aware of these stories. This is old data on the sea urchin egg from the Whitaker laboratory, where a fluorescent probe is put into the cell, the sperm is added, and this is one optical plane, and when this peak goes up, that means there's more light being emitted from a calcium-bound probe. If the calcium availability goes down, again, within the cell, the fluorescence goes down. And what you see is this rapid oscillation of calcium, these calcium-locked waves that play critical roles in waking up an egg and getting it ready for the next step after the sperm is entered, and that is to create a zygote and then an embryo. So this is a young woman that took on the challenge, Allison Kim, and she took some of the fluorescent probes I've already shown you, and she stained eggs with them, and the very first experiment she did were it lit up this little bucket here on the side of the cell. It's called a polar body. And this is it blown up. And Teresa said, congratulations, you just stained the trash can of the egg. So this is a place where the extra copies of chromatin go during the first meiotic reductive division. This is Rich On, who helped us build the microscopes that you'll see in these studies. Allison took that on and then really has dissected and really opened this up. And we got with a grant from the Keck Foundation, and then later from NIH, from Program Project Grant, NICC, and now in MITMS, R1, we've really opened up this field extensively. I'm going to tell you a little bit about the collaboration with Vinay Dravid in the engineering school, and then also with my colleagues in the developmental biology field. So I just have to introduce you to a little bit of egg biology. As the egg is just about to ovulate, it has 4 copies of maternal chromosomes. It goes through an asymmetric division to give off 2 copies to this polar body, and then it arrests and it waits for the sperm. This cell is pluripotent. It has now about 20 hours to be fertilized or die. And then once it's fertilized, the second copy of maternal chromatin is ejected. These will eventually be resorbed in the egg for their nutrient value. And then the male and the female pronuclei migrate to one another, and when they now cross over, then you have a new genetic organism. And then you finish meiosis, and then you go into mitosis. Okay? So what Allison did was measure, and I won't go into the details of how she did this, I'll show a little bit of it in the later slides. She measured in single eggs the number of zinc atoms. She counted 40 billion in this stage of the egg. And then 10 hours later, there were 60 billion. And this is reproducible, statistically clear, and has been validated by a number of other methods. So we've never seen that big of a surge in zinc over such a short period of time. And she went further, and that's a 20 billion increase in the number of zinc atoms. How much does that correspond to concentration-wise? Well, if you put it, it falls right on the line if you divide the number of moles by the volume. So the mouse oocyte has a normal amount of zinc. And so that 50% increase is then what's, I think, playing a critical role in changing the biology. She also showed that when zinc went up, right after fertilization, it started going down. It never went as low as it started. And we asked, where did that zinc go? And these are her original micrographs of time courses. And she put a calcium probe inside and then activated the eggs. They think they're fertilized, and they start their calcium oscillations, just like the sea urchin ones I showed you a few minutes ago. Then what happened is something that we still are stunned by. We began to see massive coordinated exocytosis of zinc, or what we interpreted was that. So these we called zinc sparks. And what I'm going to take the last few minutes is to tell you about how those zinc sparks work. I do want to assure you that this increase is essential. The cell spells out at telophase if it doesn't accrue those 20 billion zinc. And if you put zinc in after it started sparking and the zinc started going down with a chemical ionophore, the cell tries to go backwards in the developmental trajectory. It reforms the actin cap and it starts to realign the metaphase chromosome. So it's a fascinating thing. And it's also asymmetric. This is the zinc-starved egg that's been trying to mature. And instead of having a little sized can, it's almost gone through a symmetric mitotic division. And so these have been described. I don't want to spend a lot of time on the signaling pathways, except to point out that one of the critical regulators of the progression through the meiotic cell cycle is the anaphase promoting complex. And this is an E3 ubiquitin ligase that catalyzes the degradation of the cyclin P. So this protein, ME2, is an inhibitor of that. When it goes up, it shuts down the anaphase promoting complex. The cell cycle arrests. This ME2 protein is a ring finger protein, and both Miranda and also Tony Perry's group have done really elegant studies showing that in fact the zinc binding capacity of ME2 is critical to this function. And our model now is that the cytostatic factor that maintains eggs in this metaphase arrest until they can be fertilized is actually a zinc-dependent switch. Okay, how did the zinc get in? We have over 24 zinc transporters in mouse genome and in human genome. It's an extraordinary array. There's only a handful of iron transporters and a handful of copper transporters for 24 zinc transporters. And they move generally in one direction out of the cytosol, or they move the zinc into the cytosol depending upon the class they're in. The question is which of those were at play, and Teresa's group used morpholino injections and went through and screened a lot of these zinc transporters and shown that ZIP6 and ZIP10 are part of the maternal inheritance of RNAs, and that these two are essential to get zinc loading into the cell. So our model right now is that these are the characters that bring that 20 billion zinc atoms in, and that ME2 is one of the targets which those zinc atoms go to then shut down progression in metaphase. The interesting thing is, what are the 20 billion zinc atoms doing? And to understand this, we're developing a field of metalloproteomics. We're asking what proteins are actually unique to the M2 phase that are known zinc proteins. And 54 known zinc proteins are unique. They're not found in GV, and they're not found in the zygote stage. So these are among our candidates, a lot of ligases, histone ligase, histone E3 ligases, histone modifying proteins. And we're now sorting through and trying to identify which ones get metaloaded, what the zinc is doing, and how it's regulating. So it's going to be a complex story. I'll ask your patients as we try to work through what those receptors are and how they work. The last few minutes, I want to show you where the zinc spark comes from. To solve that problem, we had to synthesize new molecules and develop new imaging methods that actually map the location of zinc atoms within the cell. And Emily Kay developed, and when she was a postdoc in the lab, developed an extraordinary probe. This is a live egg that's staining for its DNA and for the zinc in green. And this is the M2 phase. This is the arrested egg. So what's remarkable about our probe is, first of all, it illuminated such interesting possible compartments, but it also did it at 50 nanomolar, an exceedingly low concentration that doesn't disturb the pool of zinc within the egg. And what she saw is that pattern changed from the GB to the M2 and then to the 2 cell, or to the zygote of the 2 cell. And notice that it forms this hemispherical distribution. And this is called the cortical granule-free zone. This is a type of biology that was seen before anything was known about zinc, about other types of granules that are released at the time of fertilization. And I'm not going to take you through all that biology, but I'm going to show you what that looks like when you ask, are those really zinc-loaded vesicles? And what we had to do is build a new type of electron microscope. This is really the work of Vinayak Gurjeet in collaboration on our tech grant that put element-specific detectors into the system so that you don't have to stain the sample and you can quantitatively identify compartments. And here's the kind of thing you see. This is the oplasmic membrane. This is paravillan space and the zona pellucida up here. These bright areas are electron dense, and when we take the spectrum of it, you can see clearly the presence of zinc in those blue spots, but not in the red spots. And in fact, we can go further and count the number of atoms there. We can take it to the bio-nanoprobe at the synchrotron. And this is a real opportunity, I think, for NIH, particularly NITMS, to help us open up whole new fields of biological questions and address specific issues in pathological space, because it gives 30 nanometer spatial resolution in XY, and it's set up to be able to do tomography so that we can actually develop a three-dimensional picture of at least a dozen elements simultaneously and it has cryocapacity. So I'll show you, we've done it on slices there. It's still thick. So this is the M2 egg, and the bright spots are where the zinc is really enriched. And you can see, again, that hemispherical localization. So this is another image of that. So do these vesicles really release the zinc? Are they vesicles? We think we proved that. This is a live cell with Emily's probe again on the inside, a different probe on the outside, and it's been activated, and this is about twice real time. You can actually watch the zinc fluorescence inside the cell decrease as the other probe picks up the zinc outside, and it increases. And I won't show you the time traces, but they directly correlate here. And they're always preceded by a calcium surge. So this is exactly mapping onto the physiology of synaptic vesicular release, and we think this is a really nice example of how the cortical reaction is giving rise to a massive exocytosis. Just very quickly, is this true in invertebrates and amphibians? And without going in detail through the data, we're looking at both stages of the zinc flux, the influx, and also the zinc spark, and now we've seen it in each of these organisms. I'll show you a couple highlights from this. In the worm, the zinc is essential in its diet. Its brood size disappears, and each of the eggs start to go through maturation problems as they go through the assembly line in C. elegans. In fact, the zinc content jumps as you go from that prophase, the GB stage, to the zygote, and it's an even bigger jump in zinc than you see in the mouse cells. And this is the data. This is that 20 billion atom cell. Okay, so that part is mapping onto it. There's a variety of important spindle defects when you just grow them on insufficient zinc, and we're now watching and looking for the zinc spark in the egg. In the frog egg, what you can see is the zinc spark is really beautiful. It really comes on very strongly when the cells are activated with ionomycin, or if they're fertilized with sperm. Let me see if I can get this other one to work. Just watch that one again. Let's see if this one will light up. You'll see that rapid lighting up of the entire jelly region around the egg. This is why we go into so many organisms. Well, it's not to catalog the biology, but each organism offers a great advantage in dissecting these pathways. I can do biochemistry on an egg that's 10,000 times bigger in terms of volume than the mouse egg. We can isolate the compartments and evaluate them as we go forward. So we want to know if we can use this information to really predict outcomes in IVF, and this is an application that human health is really important because multiple births that result from IVF clinics putting in multiple embryos into a woman, those multiple births, there's a significant morbidity and mortality associated, even with twins. So there's a real drive on to really detect what a good egg is, and right now there's very few methods that don't perturb the egg or the developing embryo. And so we've begun studying zinc efflux, and in fact now has shown, I won't take you through the data, that the intensity of the zinc spark is directly proportional to the survival rate of progression to blast in the mouse, and we've also now been able to look at the human egg. Now, this is where I have to step away from NIH funding. So because of the Dickey-Wicker amendment, human eggs cannot even be examined going through parthenogenesis in the lab because of the way this amendment is written. It clearly has some ethical concerns about making human life, making a fertilized egg in the laboratory. You can't use NIH money to do that, but parthenogenesis is the activation of the egg with no possibility of it proceeding to an embryo. It doesn't have the right genetic complement, and these things will not progress. But nonetheless, we're prevented from doing it. Teresa Woodruff, my collaborator, has really spent a lot of time trying to get that law changed so that reproductive research can proceed at a clip comparable around the world if we're going to solve these problems. So that the work I'm showing you here, the zinc spark that we see in the human egg, is unfortunately, there it goes. And I don't think my other movies are going to run on this platform, but you can see the zinc spark emerge. This is a human egg that's been activated that through volunteers that contributed eggs they weren't going to use in the IVF clinic. And then with support from Thering and from the Keck Foundation, we were able to do all these experiments without any type of NIH funding. I hope in the future to be able to tell you that we can make progress more rapidly, but that's going to require change in the legal code. But it's really exciting now to see that the human egg is getting sparks, and in fact, these are correlating with the very limited data we have with the successful progression to a blast stage, which would then be implanted. Okay, last couple of questions are, what is this thing doing? And I wish I had more molecular descriptions for you. One that I'm just going to show you in a minute, this is just submitted for publication, is that when the sperm fertilizes the egg, the zinc concentration in the zona pellucida right outside the egg rapidly increases to a really non-physiological concentration. It's almost millimolar right at the egg surface. And what we ask the question is, could that spark, could that be transforming the outer shell of the egg and making it less permeable to sperm? And in fact, that's exactly what's happening. First of all, we can show that the zinc content of the eggshell goes up by measuring single egg cells at the synchrotron. It goes up by about 200 to 300%. So the zinc is sticking, and when you look at the surface, this is an M2 egg, and this is after it's been activated, and now the block of parspermia is starting. You can see the difference, the thickness of these mesh-like network that is the glycoprotein matrix of zona pellucida. If you just squirt a little bit of zinc, not even going up to those high concentrations, or just almost achieving them, what you see is a significant change in the biophysics of that polymer shell, and in fact, these are statistically indistinguishable. So this is a really exciting time. I hope that through this demonstration of, and really we've spent a lot of time documenting whether this is an artifact, is it reproducible, how much of the zinc is really coming from the outside the cell, where is it going, and what is it doing. And we've really just begun to scratch the surface of those questions. The NE2 protein is, I think, is our first clue, and there's an NE1 protein that controls mitosis, and so it's possible that zinc surges are going to be involved in almost all of the mammalian cell cycle decision events, but that's a wide open question. There are zinc waves in neuronal cells when they become activated. There's zinc sparks in pancreatic beta cells when they release insulin, and this is essential to the biology. In fact, the zinc transporter is the most widely found correlative in GWAS studies with type 2 diabetes. So zinc is playing a role in metabolic disease and infectious disease. These fluxes are often part of the battleground between a pathogen and a host. And so I feel like we've just begun to scratch as a community the surface of what the movement of metal ions can do in terms of establishing the health or the death of the cell. So I want to stop there and really thank NIH and NIGMS for, without the kind of continuous support in my research program, really I don't think any of this would have come to fruition. I want to thank all the research students and postdocs who I was just barely able to mention some of them in the course of this talk. They really made these astounding discoveries and breakthroughs, and I want to thank you, Tom, for a great day here and you for your attention. So I think we have time for a couple questions.
Glutathione was kind of conspicuous by its absence. Have you looked at that as a possible chelator for zinc?
Yes. So glutathione has an affinity on the order of 10 to the minus 8 or 10 to the minus 9 for zinc, and so it is a significant binder. And it doesn't compete with the proteins, certainly the ones I showed you, or with most zinc finger protein sites. Dissociation constants for enzymes like carbonic anhydrase and carboxypeptidase and metalloproteases are typically in 10 to the minus 14th to the 10 to the minus 16th molar range. So glutathione could actually be buffering zinc when it's in a transient state, but it can't pull the zinc out. And you're right, it's very concentrated, 3 to 5 millimolar in the reduced state in most cells. But we don't think that's part of the general resting state of the pool. So,
Actually, just sorry, two questions. The first one is, during the zinc sparks, eventually zinc homeostasis has to be maintained. So how is that being taken care of? And the second question is, from the proteins that are specific to all those 54, do you have any clues as to whether there are specific zinc binding proteins that are actually carrying zinc to the vesicles?
Yes. So the last question first. Of those 54, none of them are known transport or trafficking properties. The closest is metallothioneine, and it's not one of those that has a space set differentiation. Metallothioneine gets to your first question, which is typically involved in cellular metal homeostasis, acts as a buffer as transcription ramps up very quickly when zinc goes up. And so you say, in the egg, how could it accumulate 20 billion zinc atoms without inducing metallothioneine and inducing the zinc transporters that dump it out? And we were puzzled by that too, but it turns out that the egg goes into a transcriptionally quiescent state just after GV, and it stays transcriptionally quiescent until about the 4-cell stage. So we've actually shifted to a state in cell cycle growth where the normal mechanisms that would regulate zinc homeostasis are off. But just for good measure, you might, if you know the story of the MTF1 gene that Walter Schaffner has cloned, it is an activator of metallothioneine expression when zinc goes up, but it's a repressor of ZIP10.